Welcome, everyone. First, I would like to give my sincere appreciation to the organizers for having me present today. As seen in the title, I will present several unusual product characteristics in biologics that we term unusual suspects and how mass spectrometry can reveal the nature of these unexpected results. In today's presentation, I will share several case studies from our lab highlighting on the techniques used in fully characterizing these anomalies during cell line development. Here is an outline of my talk. First, I will give a brief introduction to my group's role in large molecule early development. Next, I will describe several case studies focusing on mass spectrometry and related techniques that were used in characterizing sequence extensions, rare PTMs, and amino acid misincorporations that are otherwise elusive under standard modes of operation. Finally, I will conclude my talk by summarizing our findings and what we hope to accomplish in the future. This figure represents the general stages in large molecule early development. Once a target is declared during pre-portfolio research, candidates in the range of 100 to 1,000 molecules get screened and filters through lead generation and lead selection to less than a few dozen molecules that enters our department, the Cell and Developability Sciences, or CDS for short. The CDS group is responsible for the selection of the cell line that goes into late-stage development and is a function that sits in between discovery and development. The analytical function within CDS uses mass spectrometry to characterize molecules generated during cell line selection. These molecules include mAbs, drug conjugates, vaccines, and mostly multi-specific monoclonal antibodies used in immuno-oncology. Cell line development is depicted in this figure as a funneling process where the most desirable clone is selected based on a product quality expression, stability, and the titer, which is most important. CLD starts with the transfection of expression vector with the gene of interest into the host cell line. The single cells are next plated by isolating clones from pools using a process known as VIPS, or verified in situ plate seeding. These single clones are then expanded to 96 deep well plate screens, fed batch shake flask screens, and final amber bioreactor screens to finally select the top clones. The entire process takes roughly about 6 months, depending on the molecule, from transfection to selecting a manufactured cell. We use fit-for-purpose analytical methods, including mass spectrometric assays, for product quality analysis throughout this screening process. Our analysis starts with molecules, usually that are fully purified from cell culture. The current process is semi-automated, and liquid handling robots are used mostly for digestion and sample introduction. Standard LC-MS workflows as well as high-throughput rapid-fire MS is used as analytical instrumentation. We are also integrating sample preparation and mass spec analysis, where robotics will interface with sample preparation and mass spec analysis in the near future. Once the data is generated, our end-to-end automated data analysis pipeline using protein matrix BIOS performs protein and peptide level analysis. We have further expanded these workflows with BIOS Enterprise, which is an instance of BIOS software that resides in the Amazon workspace that can centralize the analysis with multiple users accessing, processing, and sharing the data across the organization. This brings me to the main topic of this talk, of unusual suspects in cell line characterization. In this study, we are identifying and characterizing the extension of a heavy chain of a multi-specific antibody. This work began when our colleagues from protein purification noticed an additional unresolved peak in a SEC run of a bispecific monoclonal antibody. To further understand this unknown SEC peak, fractions were collected and subjected to GX2, which is electrophoretic size-based separation, under both non-reducing and reducing conditions. The unknown SEC fraction shows additional higher molecular weight species in both chains of the map. From these observations, we concluded that further investigations are needed to figure out the nature of these unknown impurities, as impurities can affect the biological activity, efficacy, and immunogenicity. It can also lead to costs in developability and manufacturability of a biologic. We next perform native mass spectrometry to ensure that if this modification is non-covalent in origin, that we preserve it. The results from the native SCC LC-MS using high-resolution MS is shown here. The main peak corresponds to the intact mass of the bispecific MAD, which usually also contains glycosylation, while the shoulder peak shows several peaks approximately 5.5 kilodaltons heavier and having peak spacing corresponding to a sialic acid residue, which are usually positioned terminally to the glycosylation. Due to the high mass of this modification, we postulated that the impurity could be a combination of glycans, both N and O-link possibly, additional sequences, and additional post-translational modifications that could give rise to this mass. In order to localize these modifications, which are combinatorial in nature, we dissected the intact molecule by simple reduction of disulfide bonds that connects the light chains and the heavy chains together of the map. This workflow can be performed in conjunction with removal of N and O-link glycans that may provide evidence of a non-glycan modification, and reduced mass analysis performed in series with Saller-XO operator, which is an enzyme that cleaves a peptide backbone adjacent to a O-link glycan. If we see a fragment, this will inform if the modification is an O-glycan by the loss of a mass on the polypeptide sequence after digestion. The reduced mass spectra collected from the MAME peak and SCC shoulder peaks are shown here. The difference of these spectra is the addition of approximately 5.5 kilodalton peak found exclusively on both the heavy chains. The light chain has no additional masses. This led us to believe that the additional sequence variant could be present at either terminus of the heavy chains, co-modified with glycosylation. We also use a combination of enzymes in series with a protein A purification step that can further narrow down the purity modification to either the SCFE arm, Fab bomb, or the FC of each heavy chain. The two enzymes used are Fabalactica and ICE that cleaves above and below the hinge disulfide bonds respectively. However, due to the lack of consensus sequence on the heavy chain of the SCFE arm, Fabalactica cleavage alone cannot isolate the SCFE domain. The spectra shown from this analysis for the main peak and the SCC shoulder are shown here. The difference of the spectra is the presence of multiple peaks approximately 5 kilodaltons heavier found exclusively in the SCC shoulder peak that has the SCFE domain. This led us to hypothesize that the SCFE linker may be glycosylated with SCFE N-terminus having the extended sequence. To test this hypothesis, we performed the following tandem enzyme digestion experiment. Each having a protein A purification enrichment step that allows us to isolate each domain separately for peptide mapping. Trypsin and other endoprotease digestions were performed to generate peptides for each domain. Additional labeling was also performed using a hydrophobic tag known as TMPP to ensure the retention of small hydrophilic linker peptides found in the CFE linkers. These experiments did not show any evidence for CFE linkers to be modified, neither did they show the presence of the extended sequence N-terminus of the CFE. Therefore, we shifted our search of this modification to the FC domain. We want to confirm the sequence extension of the FC domain and confirm that the possibility of glycosylation is present due to the extension. The motivation for the shift in our strategy comes from the peptide mapping experiment that indicates the addition of a mass on the C-terminus sequence with a wildcard mass search. We then obtain several sequence tags downstream of that C-terminus from the canonical sequence. With the partial peptide sequence, we were then able to suspect cryptic splice sites. With these sequences, we designed primers and performed RT-PCR. Sanger sequencing of a 0.7 kilobase fragment matched a splice site on the HPRE or the hepatitis B post-transcriptional regulatory element that is used to enhance gene expression. We also performed splice site predictions and identified a splice donor and acceptor site for this splice isoform with 89% and 65% confidence respectively. This is a detailed view of the splice donor site located at the end of the C terminus. Shows alignment with the corresponding mRNA sequence and amino acid sequence. In a similar fashion, we can obtain the splice acceptor site, which is about 1.8 kilobases downstream of the C terminus on the HPRE sequence. Based on these predictions and RNA sequencing data, we in silico translated the mRNA sequence to obtain the amino acid sequence of the protein. The sequence also shows several patches of serine and threonine residues and a C-terminus cysteine that can potentially be capped with cysteine or glutathione. And the serine and threonine residues can be potential sites for glycosylation. Our database searches can now identify surrogate peptides or tryptic peptides mostly with both O-link and cysteine modifications based on this in silico prediction. The results of this experiment shows the identification of the major product with the expected C-terminus ending with the glycine residue. And PSM shows extension sequence ending with cysteine. PSMs of both the unmodified and modified sequences can be easily assigned, and modifications can be assigned to these sequences. Modifications for glycosylation shows about 20% site occupancy, and cysteinylation shows about a 95 site occupancy. We also identified many other glycosylation sites that are not actually annotated here on the other serine residues, but we lacked confidence sequence ions to confidently assign these modification sites and to predict the occupancy. We next confirmed the glycosylation site on threonine on the extended sequence where peptide mapping shows indirect evidence that the first threonine residue is glycosylated. This was done using Sialox operator where Sialox cleaves N-terminus of a glycosylated threonine. Having the unknown sequence determined by peptide mapping, we also wanted intact pep-protein level evidence. We then incorporated the new sequence mass and matched the mass of the extended splice isoform for both heavy chains by performing mass analysis of the reduced and deglycosylated map. We also confirmed the site of glycosylation of the intact map by reduced mass analysis performed in series with Sialox operator. The spectra shows an added sequence of 441 daltons corresponding to the cleavage before the first glycosylated threonine. This result correlates well with the peptide level evidence that we obtained. In summarizing this study, we have successfully identified and characterized the sequence variant using a variety of mass spectrometry tools and techniques. We have confirmed that the impurity that we observed in the SCC profile to be a sequence extension of a C-terminus FC that has 26 additional amino acid sequence downstream of the terminal glycine. We also confirmed that the serine and threonine residues are glycosylated with approximately 20% occupancy per site, and the terminal cysteine is at an occupancy of about 95%. In our next case study, we examined rare post-translational modification on an FC fusion protein. These modifications can be induced physiochemically or biochemically via enzymes. FC fusion protein is susceptible to both enzymatic and non-enzymatic clipping. They can also undergo various kinds of chemical modifications on methanines and threonine residues. Biochemical oxidation of proline and lysine residues on linker regions are also possible. Further biochemical modifications on the oxidized lysines are what we observed in this study. And finally, biochemical modification of serine residues on the linker regions can take place. The reduced mass analysis of this Fc Fisher protein showed several peaks corresponding to multiple oxidative modifications and a peak corresponding to a modification mass of approximately 340 Daltons, which was quite perplexing to us. In order to understand the high levels of oxidation, we carried out peptide mapping experiments to localize and quantify oxidation. Through these experiments, we identified oxidation propensity towards tryptophan 25, lysine 28 on the N-terminus peptide, and proline 277 on the C-terminus linker. From these sites, oxidation of proline 277 correlates well with the overall oxidation of the clones. This suggests that the overall oxidation of proline is the predominant type of oxidation, which is biochemically driven by enzymes. We next subjected the peptide data to a variety of wildcard mass searches to localize the 340 Daltons. A mass range of 335 to 340 Daltons, having increments of 1 Dalton, covered the required search space to confidently localize the delta mass with the mass measurement errors we were observing in the reduced mass data. These searches were also restricted to proline and serine residues, as we felt that these were the most likely residues that made up the two linkers. In our initial results, we identify a high-scoring peptide comprising of the N-terminus linker region having the 340 Dalton modification. However, localization of this modification was not possible due to poor coverage adjacent to the serine residue. Furthermore, if you see the two most dominant product ions in the spectrum were not assigned. We also noticed that the absence of the cleavage at lysine 28 and the same residue that's also oxidized. With these observations, we suspect that the 340 Dalton modification could possibly be associated with lysine 28. We next chained the same wildcard mass search in a second search restricted to lysine residues. This resulted in a spectrum that identified most of the peaks localized in the 340 Dalton unknown modification on lysine 28. However, the nature of the modification was still unknown. Upon close examination of the MSMS spectrum, we noticed a series of doubly charged peaks that were unannotated. We reasoned that these may be second-generation product ions produced from the unknown 340 Dalton modification. Looking at the mass spacing of the neutral masses, we manually deconvoluted the peak spacing between any two neutral loss peaks as 162 Daltons. This corresponds to the assembly of two hexose sugars that are coupled through a glycosidic bond with the loss of water. We propose that lysine 28 has the correct consensus motif of having any residue in front of a lysine and a glycine on the sequence for sequential enzymatic reactions, where the first step is the conversion of lysine to hydroxylysine, and the second step is the transfer of two galactose units to the oxidized lysine. Now, the deconvoluted centroidal delta mass corresponds quite well with this modification that we are proposing, which is a glucosyl galactosyl modification with a delta mass error of about slightly over a Dalton, which is pretty good. Since the levels of glucosyl galactosyl modification is dependent on lysine 28 oxidation, we further investigated the effect of all different types of oxidative modifications, chemical as well as enzymatic, during a bioreactor run where we are examining the oxidation levels of the clone at an initial time point, which is T1, and comparing the same clone to a later time point, which is T2. We see clear statistical significant decrease in time point T1 to T2 ratio for proline 277 lysine 28 oxidations. Also from these observations, we support the notion that both proline 277 and lysine 28 are oxidized by the same class of enzymes. And we don't see the chemical oxidations trending in this manner. When we examine the glucosyl galactosyl modification for the same clones and time points, we observe a similar trend in the modification due to the transfer of a galactose to the oxidized lysine. Only fraction of the oxidized lysine undergoes complete glucosyl galactosyl modification. The presence of incomplete hydroxylysine hexose in the latter time points may indicate that glucosidase activity may be removing the terminal sugar, as reported in the literature. The estimates of oxidation on lysine and glucosyl galactosyl modification is strongly correlated in clones examined in the two FC fusions having lysine 28. Also, the glucosyl galactosyl modification estimates are strongly correlated between peptide mapping and reduced mass analysis for both molecules. Using two different quantitative methods, we were able to prove this for reduced mass analysis. We next examine host cell proteins by peptide mapping. We use label-free quantitation to estimate relative levels of enzymes present in culture media. Deoxygenases and galactosyl transferase present in clones at early time points. This high prevalence of enzymes in cell culture supernatant further supports that sequential catalysis of this modification is happening. Finally, we see linkers made up of G4S are highly susceptible to xylose modification, which is a modification found in plants. These observations have also been reported or observed in biotherapeutics with G4S repeats that are fairly long in certain types of host cells. In order to mitigate xylosylation in non-MAP platforms, we have developed cell line knockouts of the xylosyl transferase activity. In summarizing this study, we have successfully identified and characterized a rare modification of galactosyl glucose on a therapeutic fusion protein occurring at a specific lysine residue. We also observe xylosylation of linkers that have G4S repeats by peptide mapping as a more sensitive method of detection. In our final study presented to you, we examine unusual amino acid misincorporation during translation. These variants, we believe, are happening via misacylation of a pool of amino acid variants formed by reactive oxygen species-mediated biochemical pathways. In order to look at these variants, we have developed end-to-end workflow from data generation to reporting, consisting of the BIOS software that performs extensive searches for possible sequence substitutions. The variant peptide sequence go through a rigorous decision tree in which PSMs are further validated. The nodes of the tree are critical in the confident reporting and quantitation of a variant. The retention time between wild type and variant and the predicted delta retention times between the wild type and variant are important in initial assessment and triaging of the spectra. A list of variants are then reported. The ability to simplify decision making based on a filter is then manually validated by an analyst where human intelligence and intuition becomes useful in making the final decision. A typical SVA analysis includes accurate mass assignments of fragments in a spectrum to a mass difference between a wild type and variant peptide sequence, among other criteria. In this example, commonly observed asparagine to serine variants at multiple positions on a peptide are confidently assigned, and the LC peaks are resolvable to quantify each positional variant of this particular asparagine to serine substitution. This is a typical example of how we would assign a misincorporation. In the second example, which is rather atypical, we found a phenylalanine to tyrosine variant on a single position on the sequence. However, to our surprise, we observed 3 well-resolved LC peaks of the same mass. Of these, we also obtained identical MS2 spectra for two of the 3 peaks confirming the variant, as shown in the red dot where MS2s are triggered. In addition to this peptide, we observed several other peptides throughout the molecule with a characteristic triplet of phenylalanine to tyrosine misincorporations. If we examine the MS2 spectra for these LC peaks, we can confidently localize the modification mass to the corresponding residue with high degree of specificity. With this observation, we were speculating the possibility of isomeric versions of tyrosine that can be chromatographically resolved as shown on the top. So there can be ortho-meta as well as paratyrosine. To test this idea, we synthesize the variant peptide with all three isomers of tyrosine and perform a spiking experiment to compare the original data. We confirm each peak in the original data with the corresponding tyrosine, ortho, meta, and paratyrosine isomers by matching retention times as shown here. And what's shown on the top is an equimolar mixture of all three variants. The standards also corresponds accurately to the peaks observed in the actual data as shown here. And then we look at the MS2 spectra. As seen, the MS2 spectra, the product ions are similar to each other in terms of the backbone fragment ions. This confirms that these are true isomers. Then we made use of these standard peptides to obtain response curves as shown here to quantify the ratio, quantify the isomers, ortho, meta, and para in the tyrosine variants. We then compared absolute quantification estimate with relative estimate we obtained by peptide mapping. The scatter plot on the left shows the percent variant peptide estimated for each of the three isomers as a function of the absolute amount of each clone obtained from the calibration curve. The data shows good correlation between the variant estimates of a peptide map with absolute quantitation. The scatter plot on the right shows each isomer normalized to all other isomers as a function of the absolute amount calculated by again by the slope of the calibration curve. This analysis clearly shows that the isomer levels are significantly different for the clones in this study. In particular, we note that both ortho and metatyrosine are more prevalent than the commonly observed paratyrosine variant of these peptides. In summary, we believe that the variant peptides with tyrosine isomers are formed through intracellular ROS-mediated mechanism. This observation is supported by reports of their prevalence in stress cells and in certain pathological states. We have accurately quantified isomeric variants using standards, and these estimates are in good agreement with relative variant levels estimated by relative quantitation. In conclusion, I hope you have witnessed the complexity in characterization when unexpected or unusual variants are present in biologics. We have used a variety of MS techniques to understand these anomalies. Through case studies, we examine sequence extensions by splicing during transcription, rare post-translational modifications such as galactoside glucose and xylose modifications that are atypical, and unusual sequence variants during translation such as tyrosine isomers. So, as future direction and my personal perspectives, as antibody designs become more complex and diverse, we would expect more unusual suspects to exist in our analytical data. Improved methods and capabilities that speed up characterization in early discovery without compromising quality is therefore essential. To improve quality in-depth characterization, we see opportunities for improvements in sample preparation, such as sample management and data tracking systems, integrating high-throughput mass spectrometry with robotic sample preparation, and increasing the information content of our analysis by integrating various assays in an autonomous fashion. We envision that we can innovate using novel technologies and that this will provide value in biologics characterization. With that, I would like to acknowledge all the contributors at Janssen and our research partners and collaborators. Finally, I would like to thank you for your time and I'm happy to take any questions.